Good afternoon, and thank you for joining us. Welcome to the first in a series of webinars focusing on biosimilars. This webinar will consider the basics of the regulatory system for biosimilar products in Europe and in the US, the history of approvals, and a brief overview of the patent dance in the US and clearing the way in Europe. My name is Matthew Royal, and to explore these issues over the next hour, I am joined by Taylor Wessing partner Wessing, and from Willkie, Farr and Gallagher, Heather Schneider and Dan Konstantinescu. To begin with, I will hand over to Heather to explain more about biological products and why they are different.
Thank you, Matt. I'm going to start the presentation here to just sort of level set a little bit so that we're all on the same page. And a lot of people attending this webinar today probably know this, but to make sure we have a common definition, I want to start with the basic, what is a biological product? And this definition comes from the U.S. Food and Drug Administration. And basically, it's a medical product used for the prevention, treatment, or cure of disease. And a biologic product is something made from natural sources, including, but not limited to, recombinant DNA technology. So a biologic drug product could be a virus, a toxin, a vaccine, a blood component or derivative, an allergenic product, or a protein, except for a chemically synthesized protein. As many of you may know, there's a difference physically between biologic drugs and what are traditional small molecule chemically synthesized drugs. This illustration comes from a US FDA presentation, which points out that biologic drugs typically have a high molecular weight, are made from living cells, and often their structure is not completely known. So the example here is a comparison between aspirin, a small molecule that's 180 Daltons in size, versus a monoclonal antibody, which is much, much larger. Another difference between biologics and small molecule drugs is the complicated production process. There are many different steps in producing a biological drug, and at each stage in this production process, a company may have patents. So, for example, there could be patents involving a vector that's used to get DNA for producing a protein, the cell itself that's been transfected with DNA to produce a recombinant protein, the growth medium or cell culture medium in which the cells are grown and expanded to produce the protein, and then the purification process that takes the raw output from the cells and purifies it to make a final drug product. Here in the United States, the market for biologics is robust, although not as mature as it is in Europe, as we'll talk about during this presentation. This slide lists the top 10 biological drugs in the United States and includes their global sales figures for last year. As you can see, the top-selling drug in the United States as far as biologics is Humira, which is AbbVie's adalimumab product. It had global sales last year of $18 billion. Another point that's seen on this slide is that many of the top-selling drugs are monoclonal antibodies. Those are typically indicated by having the MAB suffix in the name. And so as you can see in this list, adalimumab, rituximab, trastuzumab, and others are all monoclonal antibodies with billions of dollars in sales. I'm now going to hand it back to our friends at Taylor Wessing to discuss the regulatory system in the EU.
Thank you, Heather. So switching to the EU regulatory system. The question is, what is a biosimilar and why are biosimilars treated differently to generic products? As we've seen, biosimilars are biological products containing an active substance that has already been authorized as part of another biological product. However, biological products are complex molecules that cannot be precisely defined due to post-translational modifications which have an impact on the activity of the product. In addition, these products are produced in sensitive biological systems. Changing circumstances such as the temperature or pH only slightly might lead to a dramatic change in the characteristics of the product and its homogeneity. This is why it is often said for biological products, including biosimilars, that the product is the process. Due to the complexity of the products, the standard generic approach of establishing bioequivalence is not appropriate for biosimilars. A different approach is required to demonstrate similarity rather than equivalence, and this means more extensive evaluation of a biosimilar than of a small molecule generic. As a consequence, development periods are longer, and the cost of development of a biosimilar will be higher than for a generic product. This also means that there are likely to be fewer entrants to the market, and the discounts are typically lower than those seen for generic launches, just about 10 to 30%. This is, however, not the case for every biosimilar in every market. Switching to the regulatory framework, the basis for biosimilar applications in the EU is Article 10, Paragraph 4 of Directive 2001/83. This provision states that the results of appropriate preclinical tests or clinical trials must be provided to show similarity to a reference biological product. The type and quantity of these data are set out in the annex to the directive and in detailed guidelines. Guidelines adopted by the European Medicines Agency, the EMA, are therefore incorporated into the directive by direct reference. This provides considerable discretion to the EMA in its approach to authorizing biosimilars and means that the agency has got the flexibility to make necessary changes to the guidelines based on the experience it gets assessing biosimilar applications. Biosimilar applications must be made to the EMA. The use of the centralized procedure is mandatory. Applications require a complete quality dossier comparing the biosimilar to the reference biological product, including amino acid sequence and post-translational modifications, such as the glycosylation pattern. Then, on a case-specific basis, a preclinical program and a clinical dossier will be needed. In most cases, this requires a comparative clinical trial to demonstrate similarity. But it should be noted that this is not an absolute requirement, and in specific circumstances this may not be necessary. Although the data required for a biosimilar application differs from small molecule generic applications, the same data exclusivity periods apply to both. This means that a biosimilar application cannot be made until 8 years after the marketing authorization for the reference biological product has been granted. This 8-year period is followed by a further 2-year period of market exclusivity, during which the biosimilar cannot be launched. If the reference biological product obtains approval for an additional indication that is determined to be of significant clinical benefit, a further year of market exclusivity is also available. As we will know, this is commonly referred to as the 8+2+1 regime. As I mentioned, the contents of the guidelines adopted by the EMA are important for biosimilar applications. This slide summarizes the guidelines that have been adopted so far. On the left-hand side of the slide, you can find the guidelines that apply to all biosimilar applications. There are 3 substantive guidelines and 1 guideline setting out procedural advice, the so-called overarching guideline, then the guideline on non-clinical and clinical issues, and finally the guideline on quality issues. These guidelines were all updated a few years ago to reflect the EMA's growing experience in considering biosimilar applications since 2006. The overarching guideline explains that the guiding principle of a biosimilar development program is to establish similarity by the best possible means. This is to ensure that the previously proven safety and efficacy of the reference medicinal product also applies to the biosimilar. The biosimilar should be highly similar, and any observed differences should be justified. It is important to note that the studies conducted are not to prove efficacy per se. This has already been demonstrated for the reference biological product and need not be repeated. The goal of the studies is comparability, and the tests should be sufficiently sensitive to detect any relevant differences. To achieve this, the non-clinical and clinical guideline sets out a stepwise approach, which means that the results achieved at one stage, for instance the non-clinical in vitro stage, are used to determine the approach to the next steps, such as animal models and clinical testing. The quality guideline addresses two distinct aspects of the development of a biosimilar. Firstly, the molecular characteristics and quality attributes of the biosimilar as compared to the reference product. This includes, for instance, the amino acid sequence, which needs to be identical, but also the glycosylation pattern, which can affect the activity of the biosimilar. Secondly, the quality guideline addresses the performance and consistency of manufacturing process of the biosimilar itself to ensure that the process is robust and reproducible for each of the batches. Moving to the right-hand side of the slide, there are product-specific guidelines which address more specific aspects of particular products or class of products. These guidelines have also been updated recently. In fact, as you will see, the interferon alpha guideline is currently under review and is in a public consultation process. I will not say anything more about these guidelines for now, but would like to mention that one addresses monoclonal antibodies. Given the importance of this class of biosimilar, as we have heard, I will hand over to Matt to highlight some of the key aspects of this particular guideline.
Thank you, Manya. The biosimilar monoclonal antibody guideline applies, as the title suggests, to all biosimilar monoclonal antibodies and also fusion proteins. Because it will apply to so many more biosimilars than the other product-specific guidelines, due to the number of monoclonal antibodies on the market, it's worth spending a few minutes considering this guideline in more detail. The guideline was first adopted in 2012, but it was not adopted without controversy. Amongst other things, it was said that the number of different monoclonal antibody products called for product-specific guidelines for each of those monoclonal antibodies, rather than a class-specific guideline. The guideline was, however, adopted and has been applied by the EMA to a number of biosimilar monoclonal antibody applications since 2012. The details of the guidelines set out similar principles to the overarching guidelines that Manya has discussed. The overall aim of an application is to establish comparability between the biosimilar monoclonal antibody and the reference monoclonal antibody. The way in which this is done, as again Manya has explained, is to take a stepwise approach. For clinical trials, the guideline again highlights that the trials are aimed at comparability and not at demonstrating efficacy per se. Finally, extrapolation to other indications that have not been directly studied in a clinical trial is possible with appropriate justification. Again, this allows a biosimilar to get to market with fewer studies involving fewer individuals. To give these concepts some more context, I will now turn to consider an example of how the monoclonal antibody guideline was applied to a particular product. The example is infliximab. It was the first monoclonal antibody biosimilar to be authorised in Europe. The marketing authorisation was pending while the monoclonal antibody guideline was being finalised and was granted in 2013, very shortly after the guideline was adopted. Based on this timing, it's possible to speculate that the EMA's experience in considering the infliximab application was being used to finalise the guideline. And so it's interesting to consider some of the issues that came up during the infliximab application, as reported in the European Public Assessment Report. Infliximab is a monoclonal antibody to TNF-alpha. The reference biological product was Remicade, and the two biosimilar monoclonal antibodies were assessed, Remsima and Inflectra, marketed by Celltrion and Hospira respectively. The applications for Remsima and Inflectra were considered as duplicate applications by the EMA because both biosimilars were manufactured in the same facility but were marketed separately. Infliximab is indicated for a number of different indications: rheumatoid arthritis, ankylosing spondylitis, psoriatic arthritis, Crohn's disease, and ulcerative colitis. It is therefore a good example of a product where extrapolation of indications might be relevant. First in the stepwise approach, however, is the in vitro stage. In the case of infliximab, there was a difference identified in a post-translation modification of the biosimilar, which caused a lower level of antibody-dependent cell cytotoxicity in the most sensitive in vitro model of Crohn's disease. This difference was justified, as required by the guideline, by evidence that the difference was not clinically relevant. This included evidence that any differences were abolished in vivo, and also by using alternative in vitro models that were more clinically relevant and that demonstrated no differences between the biosimilar and the reference monoclonal antibody. Next in the stepwise approach, a small number of pharmacokinetic, toxicity and immunogenicity studies were carried out in rats. The pharmacokinetic parameters were similar and no difference in toxicity or immunogenicity between the biosimilar and the reference monoclonal antibody were detected. So let's head to the final step, which is the clinical program. It consisted of 2 pivotal trials in rheumatoid arthritis and in ankylosing spondylitis. Pivotal trials, however, were not conducted in Crohn's disease or ulcerative colitis, diseases which are collectively referred to as inflammatory bowel disease. Despite this, a full label was approved, including these indications. This approval was based on extrapolation of indications and was justified by additional information provided by the applicant. The additional information included a literature review, relevant in vitro experimental models of inflammatory bowel disease, genetic immunological evidence, as well as some preliminary data from a few patients, actually 23 patients with Crohn's disease and ulcerative colitis. In addition, the applicant agreed to conduct further clinical trials in Crohn's disease post-approval. Ultimately, the EMA accepted this evidence, and the infliximab biosimilars have now been on the market since 2014 and used successfully in all indications. In fact, the NORSWITCH study, published recently in The Lancet, provided evidence that switching patients from the originator infliximab to one of the biosimilars was not inferior to continuing treatment on the originator. This provides good support for the EMA's approach as outlined in the monoclonal antibody guideline. I'll now hand over to Heather to discuss the regulatory system in the US.
Thank you, Matt. So we'll talk a little bit here about the statutory and regulatory framework in the U.S., which is in much more of a state of flux than it sounds like some of the regulatory system in Europe. So first, here, as some of you may know, the production of biosimilars is governed by the BPCIA, the Biologics Price Competition and Innovation Act, that was signed into law in March 2010 as part of the Affordable Care Act. And the goal of the law was to establish a biosimilars pathway that would balance both innovation and consumer interest. And the idea of the act is to encourage biosimilar products to come to market to enhance competition, leading to better patient access and, hopefully, lower cost. To get this balance between innovator drugs and biosimilars, the BPCIA provides a period of exclusivity for reference products, followed by an abbreviated approval pathway for biosimilars. In some ways, it's similar to the Hatch-Waxman Act that some of you may be familiar with respect to small molecule drugs. However, the timing here is different. A reference product may be listed, and 4 years after that, a biosimilar application can be filed. 12 years after listing of the reference product, a biosimilar application can be approved. The idea being that you have a 4-year period of data exclusivity, and then an 8-year period where the biosimilar application can go through the regulatory review process, and also the parties can engage in the patent dance and litigations that my colleague Dan will talk about later. In order to get approval for a biosimilar, you have to show that your product is highly similar to and has no clinically meaningful differences from the reference product. Demonstrating biosimilarity usually addresses 4 factors: the mechanism of action of the drug, the route of administration, dosage form, and strength, conditions of use in the proposed labeling, and the manufacturing, processing, and packaging procedures. When preparing a biosimilar application, you can leverage the FDA's finding of safety and effectiveness for the reference product, but you still have to submit a significant amount of data. As shown on the next slide, and as you heard a little bit in the European process, the FDA here also follows what's called a stepwise approach to approval. And they look at the totality of the evidence to demonstrate biosimilarity. And this pyramid comes from an FDA presentation explaining this stepwise approach. At the bottom, you have a large body of analytical data and studies that have to be submitted to show high similarity to the reference product. Then above that, in the pyramid, you need to submit non-clinical data, similar to what Matt referred to for infliximab, some kind of animal studies and other studies, including, for example, an assessment of toxicity. Then a biosimilar developer has to submit 1 or more clinical studies to demonstrate safety, purity, and potency in 1 or more of the conditions for which the reference product is listed. And you heard a little bit about that in the infliximab study. These can include immunogenicity assessments, pharmacokinetic studies, pharmacodynamic studies, and possibly switching studies, as Matt had referred to. Now I'm going to hand this over to Matt to talk a little bit about the history of some actual approvals and launches of biosimilar drugs.
Yeah, thank you, Heather. I will take this on.
Sorry, Manya.
It's no problem. And talk first about the approvals that we have had so far in the EU. So moving to the next slide. The first biosimilar is somatropin, was authorized in Europe in 2006. Since then, as set out in the slide, as you can see, the EMA has authorized a total of 48 biosimilars. Initially, this was limited to smaller and relatively simple biosimilars, such as I said, somatropin, EPO, and filgrastims. But, as explained earlier, once the monoclonal antibody guideline had been adopted 6 years ago, a number of monoclonal antibodies have been approved. Since the approval of infliximab, there have also been authorizations for rituximab, adalimumab, bevacizumab, and trastuzumab. You can see this at the lower end of the slide. As I'm sure that you're aware, this is a very active area, so it is also worth noting that there are more biosimilars in the pipeline. 3 have received a positive opinion from the CHMP. 1 more adalimumab biosimilar, and the first 2 pegfilgrastim products. These will be authorized shortly, and other applications are being considered by the EMA at the moment. In terms of biosimilar launches, most of the approved products on the slide have been launched, and so it's easier to highlight those classes that have not yet been launched. The teriparatide products, that's the 4th line from the bottom, have not yet been launched. And in terms of the monoclonal antibodies, adalimumab and bevacizumab are still protected by an SPC, and therefore these biosimilars have not yet been launched. While it is fair to say that the European system is more established than the US, there's a huge amount of activity at the moment, and I will hand over to Heather again to discuss the history of approvals in the US.
Thank you, Manya. As you all will see, there's a big difference in the number of approvals of biosimilars in the US than the slide that was just shown for Europe. The market is much less mature for biosimilars. And basically, the U.S. Food and Drug Administration has approved 12 biosimilars, and only 4 of them have launched. So these 12 biosimilars have been approved between 2015 and July of 2018, and 4 of them have launched by now. And a lot of people in this area may wonder, why is that occurring? Some of it has to do with patent issues, some of it has to do with market competition and other issues. People at the FDA have also wondered how to make the numbers on this slide look a little different. And so this summer, the U.S. came out with a Biosimilars action plan from the FDA. It's a 4-part plan, and we'll all be watching closely how it plays out. It has 4 sections, the first being a goal to improve efficiency of biosimilar development and approval. So, for example, the FDA is going to come out with application review templates, and they're going to create a new Office of Therapeutic Biologics and Biosimilars. The second goal is to maximize scientific and regulatory clarity for biosimilar development. As you saw in Europe, there are a lot of guidances available. Here in the United States, there are very few, if any, final guidances on the regulation of biosimilars. So one of FDA's goals is to update their draft and final guidances. They're also preparing a proposed rule for notice and comment regarding the definition of a biological product. And they also plan to enhance what's called the Purple Book. It's similar a little bit to the Orange Book some of you may have heard of in Hatch-Waxman litigation, but it doesn't list all the patents that might cover a drug. It just basically lists products and their approval dates. The third goal of the Biosimilar Action Plan is to develop effective communications among patients, clinicians, and payers. The idea being to educate the public and doctors that biosimilars are safe and effective and comparable to the reference-listed products. The fourth and final topic of the Biosimilars Action Plan is pretty interesting and will definitely be the subject of discussion at our next 2 webinars on this topic. The FDA wants to support market competition by reducing what they call the, quote, gaming of FDA requirements. That's going to be a very, I'm sure, interesting and controversial area to follow. And in the next few webinars in our series, we're going to talk a lot more about what's actually going on in the marketplace for biosimilars and what some of the obstacles are to competition in addition to patents. Now, moving on, for those of you who are attending from the United States and would like CLE credit, this is the code that you need to write down, WF&G 1311. We'll follow up with you to make sure we submit your requirements. We have approval for New York, and we have approval pending in California and Illinois. And if you have any additional jurisdictions, just let us know, and we'll work on that. Now I'm going to hand off to my colleague Dan to talk about the patent dance.
Thank you, Heather, and hello, everyone. I will spend the next 15 minutes or so providing a high-level overview of the framework under the BPCIA for initiating patent litigation between a reference product sponsor and a biosimilar applicant, which, as many of you probably already know, has been affectionately nicknamed the patent dance. So the patent dance consists of a series of steps that are illustrated in the flow chart on this slide. And the steps require the applicant and the reference product sponsor to exchange various types of information in the hope of reaching agreement on a number of patents to be litigated in a first wave of litigation. The framework also contemplates a second wave of litigation for additional patents at a later date, as I'll discuss in a little bit. Now, the overall purpose of the patent dance is to help the parties narrow the issues and initiate patent litigation in a timely fashion so that there could be some or complete patent certainty by the time that the applicant is ready to launch its biosimilar product. In the next few slides, I will go over the main steps in the patent dance in a little bit more detail. So the patent dance framework is described in 42 U.S. Code Section 262. And paragraph L2 prescribes the first substantive exchange of information in the patent dance, which requires the applicant to provide certain information to the reference product sponsor. Specifically, it requires the applicant to provide a copy of the application submitted and such other information that describes the applicant's manufacturing process. It also states that the applicant may provide additional information requested by the reference product sponsor. Now, if the applicant does not provide the information in paragraph L2, the reference product sponsor can basically initiate patent litigation. It may bring an action for declaratory judgment of infringement, validity, or unenforceability for any patent that claims the biological product or that claims a use of the biological product. It can also sue for infringement under Section 271E over patents that could be identified in what is called a 3A list, which I'll describe in the next slide. Now, one thing that has played out in the courts in the U.S. Is the issue of whether the reference product sponsor can enjoin or compel the biosimilar applicant to produce its information, its application and manufacturing information. In other words, can it compel the biosimilar applicant to initiate the patent dance? And this has made it to the Supreme Court in Sandoz v. Amgen. And the Supreme Court held that there is no federal law under which a reference product sponsor can enjoin a biosimilar applicant to disclose the information. As a corollary, the Federal Circuit Court of Appeals subsequently held that the BPCIA preempts state law, so there is no state law remedies that a reference product sponsor can use to enjoin the applicant to produce that information either. So basically, the only option for the reference product sponsor is to initiate litigation. Now, if the applicant provides the information under paragraph L2 and initiates the patent dance, then the reference product sponsor is required to produce information under paragraph 3A. Specifically, paragraph 3A requires the reference product sponsor to provide a list of patents for which a claim of patent infringement could reasonably be asserted. It also requires the reference product sponsor to identify the patents on that list that it is prepared to license to the biosimilar applicant. So what goes into determining which patents to list? Obviously, that depends on the particular circumstances of the case. But as far as the standard of could reasonably be asserted, the Federal Circuit has described that it merely requires the sponsor to list patents that it believes could reasonably be asserted. And it also explained that there is no sanction for holding or asserting a mistaken belief in good faith. On the other hand, under Section 271E, the reference product sponsor could potentially waive its right to sue for infringement over any patent that should have been included in the 3A list, but was not timely included. So practically, the reference product sponsor is very motivated to liberally include patents in the 3A list. And in most BPCIA cases so far, that is exactly what has happened. The reference product sponsor has, in fact, asserted or included numerous patents covering various areas of subject matter. As mentioned earlier, biologicals are more complex than small molecules, and their processes of manufacture are more complex. So it is not uncommon in these BPCIA patent dance exchanges to see patents not only to the biological itself, but also patents related to the manufacturing process for the biological product, including patents to cell culture methods, patents to protein purification methods, cell culture media, patents to cell lines, as well as patents to methods of use such as methods of treatment. And that is that one difference between the BPCIA litigation and Hatch-Waxman small molecule litigation is that the scope of the subject matter being litigated for one application can be very broad and encompass numerous areas of technology. So in response to the 3A list of patents, paragraph 3B requires the biosimilar applicant to provide certain types of information. First of all, it states that the biosimilar applicant may provide a list of patents for which it believes the claim of patent infringement could reasonably be asserted. It requires the biosimilar applicant to provide a detailed statement that describes the factual and legal bases that each patent listed in the 3A list is invalid, unenforceable, or otherwise will not be infringed. Basically, non-infringement and invalidity contentions. Now, alternatively, if the applicant does not want to provide contentions for one or more patents in the 3A list, it can instead provide a statement that the applicant does not intend to begin commercial marketing of the biological product before the date of expiration of any patent on the 3A list. If the applicant fails to provide its 3B list or otherwise comply with these provisions, the reference product sponsor may bring an action for declaratory judgment of infringement, validity, or unenforceability for any patent included in the 3A list. So in response to the 3B list, the reference product sponsor must also essentially provide its infringement and validity contentions. Specifically, paragraph 3C requires the reference product sponsor to provide a detailed statement that describes the factual and legal bases of its opinion that the patents it listed in the 3A list will be infringed, and also to provide a response to the statement concerning validity and enforceability that was in the applicant's 3B statement. Now, the rationale behind these exchanges is that once these contentions have been exchanged, the parties should have a better idea and a better understanding of which patents are really relevant and which patents are not relevant to the applicant's biological product and its manufacturing process. And that's important because in the next step of the dance, under paragraph L4, the parties are required to engage in a period of good faith negotiations in order to try to agree on which patents shall be the subject of an initial wave of patent litigation. If the parties agree, the reference product sponsor brings an infringement action for each patent that the parties agree to. If the parties do not agree, then the parties must undergo an exchange process where each party provides a list of patents that it believes should be included in a first wave of litigation. And the reference product sponsor can then initiate an infringement action for each patent in those lists. And it's a little bit more nuanced than that. Basically, the biosimilar applicant first provides to the reference product sponsor the number of patents that it will include on its list. Then the parties simultaneously exchange lists, and the reference product sponsor cannot include more patents than the number of patents that the biosimilar applicant said it will include. So if the biosimilar applicant tells the reference product sponsor that it will include 5 patents, both parties can each include 5 patents, and the reference product sponsor can then bring an infringement action on each unique patent. The only caveat to that is that if the biosimilar applicant says that it will not list any patents, then the reference product sponsor can identify one patent and initiate litigation on that patent. So what's interesting is that the biosimilar applicant actually has a lot of control over the scope of this first wave of litigation because it can basically determine how many patents will be included in this wave. Now, if the biosimilar applicant fails to fulfill these exchange requirements, then the reference product sponsor can again bring an action for declaratory judgment of infringement, validity, or enforceability for any patents included in its 3A list. So the provisions I discussed so far basically deal with narrowing the issues in dispute and getting to this first wave of litigation, which should really include the core patents and the patents that are most relevant to the biosimilar applicant's product and manufacturing process. I mentioned earlier that there is also a second wave of litigation contemplated in the BPCIA, and I'll describe how that becomes initiated. That is basically triggered when the biosimilar applicant provides its notice of commercial marketing. And to take a step back, the BPCIA requires the biosimilar applicant to provide notice to the reference product sponsor not later than 180 days before the date of first commercial marketing. Basically, it cannot launch its product until 180 days after it provides this notice. Now, in the Sandoz v. Amgen case that I mentioned earlier, another issue related to this provision was before the court. And the issue there was whether the applicant must wait to get FDA approval before providing notice of commercial marketing. And the Supreme Court held that the answer is no, the applicant may provide notice of commercial marketing prior to FDA approval and really at any point during this process. Now, once that notice is provided, then the reference product sponsor can file a preliminary injunction on any patent listed under paragraph 3 and not included in the paragraph 5B exchange lists that I mentioned before. Similarly, the reference product sponsor can also bring an action for declaratory judgment over the same patents. Basically, the patents that were listed in the 3A list but were not litigated in the first wave of litigation. Now, here again, because the applicant controls when it will provide notice, it effectively controls when the second wave of litigation will begin. And why have two waves? I think part of the rationale is preservation of the parties' resources, preservation of judicial resources in the initial wave. It's obviously a lot easier to litigate a handful of patents than to litigate 20 or more patents. And also, achieving certainty on the core patents, the most relevant patents, might make settlement discussions easier. It might obviate the need for a second wave of litigation entirely and forgo all those expenses. So, to conclude, I think the patent dance is a fairly complex framework for initiating patent litigation. As I mentioned earlier, there have been disputes over what certain provisions actually mean, and some have made it to the Supreme Court, which has provided some clarity. But we think moving forward, we anticipate that there will continue to be disputes in the courts over the next several years as the parties, both reference product sponsor and biosimilar applicant, continue to find additional ambiguities in the law, and until those ambiguities get ironed out. So that is a high-level overview of the patent dance under the BPCIA here in the U.S. And now I'll hand over the presentation to the presenters from Taylor Wessing to talk about the legal landscape in Europe.
Thank you, Dan. So moving back to the position in Europe. Unlike in the US, there is no linkage between the regulatory system and patent litigation, and there is no patent dance in the way that Dan has just described. So what does this mean for a company looking to launch a biosimilar in Europe? Well, arguably, it actually makes the position more complicated. In order to launch, a biosimilar manufacturer must first identify any patents that are relevant to its product. Once any relevant patents have been identified, it is necessary to decide on the best strategy to clear the way. This strategy will require litigation to be brought country by country, and so will require an understanding on the part of the biosimilar manufacturer of the litigation procedures in multiple launch countries, and then to balance those litigation procedures with the commercial importance of the particular country. Now, these are fairly complex considerations, and I'm not going to go into them in detail in the final few minutes of today's webinar, but I will comment on some trends that we are seeing in Europe in patent litigation. First, it is common in biosimilar launches for multiple parties to oppose relevant biological patents in post-grant opposition proceedings at the EPO. Opposition proceedings offer a potential solution to having to litigate simultaneously in many different countries, and so are an attractive option. However, oppositions can take many years to be finally decided on appeal, and so may not provide the risk-free launch that a biosimilar manufacturer is looking for. Filing an opposition is therefore often part of a multi-pronged approach to clearing the way, and national litigation is also often required. Of the monoclonal antibodies listed on the slide, all have been subject of litigation in the UK, and trastuzumab, rituximab, and adalimumab have been opposed by multiple parties in opposition proceedings at the EPO. I wanted to draw out 2 points from these cases. The first is the relative balance between the regulatory position and litigation. There have been 3 decisions in the UK in relation to patents covering aspects of trastuzumab. The first of these decisions was as long ago as 2014, but trastuzumab biosimilars have only been authorised and launched this year. This demonstrates the potential difficulties balancing the regulatory and litigation timing for a biosimilar manufacturer. In the case of trastuzumab, it seems that the regulatory aspects rather than the litigation aspects were the rate-limiting step for the biosimilar manufacturers. The second trend is the scale and changing nature of the patent portfolios protecting a biological product. In relation to adalimumab, AbbVie have a large number of patents, as one might expect for such a large product. A number of those patent families include pending applications. And this leaves the biosimilar manufacturer with a problem, because it is not possible to revoke pending applications, and there is always the risk that they will grant shortly before launch and so disrupt the launch. A possible solution is developing in some jurisdictions across Europe, and that is to seek a declaration that a product lacks novelty or is obvious at the relevant date of the patent. This provides a defence to infringement, because either a patent is not infringed or it is infringed but is invalid because the biosimilar product was either not novel or obvious. Fujifilm successfully obtained such a declaration against AbbVie in the UK last year. And while this is an important trend, there are a number of questions outstanding in relation to such declarations, not least where in Europe they might be available. So far, for instance, declarations have only been granted in the UK and the Netherlands. And so this approach of so-called arrow declarations may ultimately have limited utility, and other strategies might also be needed. In any event, it will be interesting to see how these trends develop as more and more biosimilars are approved and launched in the coming years. So that concludes our webinar. It remains for me to thank our speakers, Manja Epping, Heather Schneider and Dan Konstantinescu, and to remind you about the second and third webinars on the 27th of September, when we'll be looking at interchangeability, substitutability and advertising of biosimilars, and on the 10th of October, when we'll look at pricing and reimbursement and the competitive landscape. For now, thank you for joining today's webinar, and could I ask you to hold on for a few moments to answer a short feedback form.